Hello students, this is your Prasanna ma'am from NEET World. How are you all? Hope you are all doing great, right? So now we are here to discuss about most interesting and important topic from the chapter Principles of Inheritance and Variations. Those are Mendelian disorders and chromosomal disorders. Most of the students will feel difficult in answering this set of questionnaire from Mendelian disorders and chromosomal disorders, right? Stay with me for this 30 to 40 minutes to make this concept very easy and on your fingertips, right? So yes, what are genetic disorders? Genetic disorders are nothing but the disorders or diseases which are caused due to abnormality in either nucleotide sequence or in the gene sequence or due to chromosomal aberrations. Genetic disorders are caused due to abnormality in either gene sequence or nucleotide sequence or chromosomal aberrations or what ma? Chromosomal aberrations. So yes, genetic disorders are again broadly classified into two types based on Mendelian inheritance. Whether the diseases are following Mendelian pattern of inheritance, then they are kept under Mendelian disorders, or if they are only caused due to chromosomal aberrations, then they are kept under chromosomal disorders. Mendelian disorders are again broadly classified into autosomal disorders, autosomal and allosomal. Autosomal again of two types, they are autosomal dominant and autosomal recessive. Allosomal again they are sex linked, right? Allosomal diseases are also called as sex linked diseases. They can be X linked diseases or Y linked diseases, or what ma? Y linked diseases. X linked diseases can either be X linked dominant or X linked recessive. X linked dominant or X linked recessive. So when they are following Mendelian pattern of inheritance, all the dominant diseases or the dominant traits are expressed both in the homozygous and heterozygous condition. Yes or no? All the dominant diseases or dominant diseased traits are expressed both in homozygous and heterozygous conditions. The recessive diseases are expressed only in the homozygous conditions. They are unexpressed in the heterozygous conditions. Keep it clear in your mind, okay? And now, amongst all these types of Mendelian disorders, what types of disorders we need to focus for our exam? Of course, from autosomal dominant, we have myotonic dystrophy. From autosomal recessive, we need to learn about color blindness and hemophilia. When it comes to X-linked, from X-linked recessive diseases, you have to learn about sickle cell anemia, phenylketonuria, Thalassemia and sickle cell anemia, phenylketonuria, thalassemia and cystic fibrosis. So yes, from X-linked recessive, you have to learn about color blindness and hemophilia. Autosomal recessive diseases include sickle cell anemia, phenylketonuria and thalassemia. So yes beta, Mendelian disorders are of two types, autosomal and allosomal. Autosomal disorders are of two types, autosomal dominant and autosomal recessive. Autosomal dominant myotonic dystrophy and autosomal recessive diseases, where you need to learn about all these diseases. What are they? Sickle cell anemia, phenylketonuria, thalassemia and cystic fibrosis. X-linked or sex-linked diseases include X-linked, Y-linked diseases. X-linked dominant, X-linked recessive diseases include color blindness and hemophilia. They include what ma? Color blindness and hemophilia. These two diseases you have to learn under X-linked recessive. So when it is X-linked recessive, in females they are expressed in only homozygous condition. In males, if the disease is on X chromosome, then they are affected readily. Okay, so yes, X-linked recessive diseases in females, they are expressed in homozygous condition. In males though, if the disease is on X chromosome, then definitely they are affected. Clear ma? So this is what you have to learn under Mendelian disorders. From chromosomal disorders, you have to look into Down syndrome, Klinefelter syndrome and Turner syndrome. Now let us understand Mendelian's disorder one by one. Okay, yes, Mendelian's disorder, those are sex-linked diseases, right? Mendelian's disorders, they are mostly caused because of mutation of a single gene or a single nucleotide sequence. Mostly they are caused because of gene mutations. Mostly they are caused because of what ma? Gene mutations or alteration in a single nucleotide sequence. So yes, because of this Mendelian disorders occur and they follow Mendelian pattern of inheritance. Okay, law of dominance, law of segregation, law of independent assortment, all these principles are followed and the traits are followed in the successive generations by the disorders which are following Mendelian principles. Okay, so they are said to be Mendelian disorders. And the first sex-linked disorder we are learning, sex-linked disorder, which is X-linked recessive disorder, that is color blindness. People who are affected with color blindness on the X chromosome, the gene which due to the mutation in a single gene in the X chromosome, they cannot differentiate between the colors of red and green. They cannot differentiate between the colors of red and green because the mutation of the gene which is present in the X chromosome will affect the cones which are coding for red and green color. So they cannot distinguish between red and green colors. So yes, this is a sex-linked recessive disease and caused by the deficiency in either red or green color of the eye, and this causes inability to distinguish between red and green colors. This deficiency is caused by mutation in one of the X chromosomes. This is caused by mutation in what ma? One of the X chromosomes, and they follow crisscross pattern of inheritance. What pattern of inheritance they follow? Crisscross pattern of inheritance. What is crisscross pattern of inheritance? The disease is passed from grandfather through daughter to grandson. The disease is passed from grandfather through daughter to grandson. Okay, and the disease is passed from grandmother to son, then to granddaughter. This pattern of inheritance is said to be crisscross pattern of inheritance, and the color blindness disease follows this pattern of inheritance. You have to fix it in your mind. Many a times the questions are asked, and it affects around 8% of males and just 0.4% of females. Why the percentage is more for males? Because if one X chromosome is affected, then they are affected. Females in the heterozygous condition, they don't show any effect because this is recessive, X-linked recessive disorder. Only in the homozygous condition they show the effect. In heterozygous, they are considered as carrier females. Okay. Now, what types of questions can be expected from this set of the concept. Okay, let us look into all the combinations of crosses that we can do. First is affected mother when crossed with unaffected father. Okay, in this case affected mother, then both the X chromosomes are predicting the disease. They are colorblind. Okay, both the X chromosomes are homozygous for the disease. Unaffected male, it is just XY. Okay, so what are the offsprings or the F1 generation we can expect is first XCX, XCY. Yes, you also do parallelly, students, along with me, and XCY. So if you look into it carefully, all the affected mothers are producing affected sons. Affected mothers gives rise to affected sons. Okay, and affected mothers are also giving rise to carrier daughters. They are also giving rise to what, ma? Carrier daughters. Okay, so all the sons of affected mother are affected. All the daughters of affected mother are carriers. You have to remember it for sure. This type of questions only you will get in your exam. Second set of cross, if we do carrier mother with unaffected father. Carrier mother with unaffected father, wherein carrier mother means it is heterozygous, right, and unaffected father it is XY. Clear, ma? So if you look into it, the daughters which are formed as a result of this are XCX, XCY, then from carrier mother 50% of the sons are affected. 50% of sons are affected. Clear, beta? From carrier mother 50% of the sons are affected, 50% are unaffected. Don't look into the complete offsprings. I am only talking about sons. When you consider the sons of F1 generation, only 50% probability is there where they can get affected. If you look into the daughters, 50% of the daughters can be carriers. 50% of daughters can be carriers. Okay, this set of crosses are applicable for two types of diseases. What are they, ma? Color blindness and hemophilia. Many a times the questions are asked from color blindness and hemophilia only, which are X-linked recessive disorders. Okay, and third set of cross, if we do third set between unaffected female and affected male. Unaffected female and affected male. What offsprings can be get? X, XC and XCX, 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 and then XY, XY. Clear, ma? So yes, all the daughters of affected father are carriers. Okay, all daughters of affected father. From this cross, what can we write? All daughters of affected father are carriers. Okay, and last set of cross, if we do, that is in between carrier mother and affected father. What is the last set, beta? Last set is between carrier mother and affected father. If in this cross you will learn that XCY, You also do parallelly with me beta. Yes, XY. So yes, all the daughters of carrier mother and affected father, 50% of the daughters are affected, 50% of daughters are affected and 50% are carriers. 50% are affected and 50% are carriers. Same way, 50% of males are affected and 50% are unaffected. 50% of males are affected and 50% are unaffected. Clear? So yes, from the X-linked recessive disorders, what type of questions you can answer, what type of questions you will get. One set, that affected mother and unaffected father, wherein from affected mothers all the sons are affected and all the daughters will be carrier daughters. Okay, this is first set you have to note. Second is from carrier mother. From carrier mother, 50% of the sons are affected and 50% of daughters are carriers. And third set, wherein affected father will give rise to all the daughters who are carriers. Affected father will give rise to all the daughters who are carriers. And last set, carrier mother and affected father will give you 50% of the daughters are affected and 50% are carriers, and 50% of the sons are affected, 50% unaffected. If you want, you can take a screenshot of this. This is very, very important. Okay, the same thing you can apply for hemophilia as well. Now let us look into the second type of X-linked recessive disorder, that is hemophilia. Hemophilia is also called as bleeder's disease beta. Hemophilia is also called as bleeder's disease. This is because of absence or mutation of any of the one clotting factor. Absence of mutation or mutation of any one of the clotting factor will not allow the blood to clot whenever the injuries occur. Whenever injuries occur, this due to the mutation of the clotting factor, clotting will not occur. This disease is called as bleeder's disease. This you will also call as hemophilia. This shows transmission from unaffected carrier mother to some of the male progeny. Unaffected carrier female, just now we have seen, no, carrier female can give rise to 50% of the affected sons. Okay, 50% of the sons are affected. Due to the presence of defective form of blood clotting factor, any one of the blood clotting factor, exposed blood of the affected individuals fails to coagulate, so that continuously the blood will bleed from the affected areas. And this results in the people bleeding for longer time after the injury, easy bruising, and an increased risk of bleeding inside joints and brain also, which is lethal. Okay, which is lethal. Next, the possibility of female becoming hemophilic is extremely rare because homozygosity of hemophilia. Okay, homozygosity of hemophilia will cause death of the embryos at the early stage of its life only. Okay, so females being hemophilic is extremely rare because it should be homozygous, and females to be homozygous, the mother should be affected and father also should be affected, or the mother should be a carrier and father also should be affected. In that condition only homozygosity of X chromosomes being hemophilic will occur and hemophilia will be in females. Okay, and this is not at all possible. If possible also, they are lethal at the embryonic stage only. Next, yes, so this is the normal blood vessel, this is the normal blood vessel, and bleeding starts. Hemophilia, if you see the first incomplete fibrin clot, continuous bleeding occurs because of the incomplete fibrin clot, and continuous bleeding occurs here, and completed fibrin clot. This is the fibrin clot. This is incomplete fibrin clot, due to that bleeding occurs. If fibrin clot is taking place, then bleeding will not occur. If they are hemophilia, clot will not take place at all. Okay, yes. Now, if you look into hemophilia. It is also classified into two types based on the clotting factor which is mutated ma. Hemophilia A and hemophilia B. Hemophilia A, it is because of mutation in the clotting factor or low amount or the absence of the clotting factor 8, absence of the clotting factor 8, which is also called as anti-hemophilic globulin protein, anti-hemophilic globulin protein. Okay, when this is affected, this will not release the clotting factor, thereby clotting will not take place. Okay, this is hemophilia 1. Majority of people are subjected to hemophilia A only. And hemophilia B, this is due to the absence or lower levels of the clotting factor 9, which is also called as plasma thromboplastin. Okay, this is also called as Christmas factor. This is also called as what ma? Christmas factor. So either of the factors are affected and clotting will not take place, thereby bleeding from the injured areas will be continued, and the severity of disease can be lethal as well. Okay, and the combination or the sets of crosses are same as we have discussed in the color blindness. Okay, same applications you can follow for this disease as well. Now if you look into the basic pedigree of this disease is first diagnosed in the family of Queen Victoria. Okay, this disease is firstly diagnosed in the family of Queen Victoria, who is the Queen for England. Okay, so yes, as she is the carrier mother has in the first generation affected son is observed. Okay, affected son is seen. Okay, and carrier mother is given rise to all the carrier daughters. You know the notations of pedigree, right beta? If this is colored, this affected, male is in square, colored, then it is affected, uncolored normal, female if it is colored it is affected, and dot at the center will resemble what? Carrier female. Circle is what? Female. Okay, these are the notations for pedigree you have to remember. And carrier mother will give rise to affected son. Okay, and carrier daughters. Carrier daughters from the first generation are giving rise to affected sons of second generation. Can you see that? Carrier daughters of first generation are giving rise to affected sons of second generation, if you can see here. Okay, this you have to remember for sure. Okay, and affected fathers will always give rise to carrier daughters. Affected father will always give rise to what ma? Carrier daughters. This you have to remember. Carrier mothers giving rise to affected fathers giving rise to carrier daughters. This you have to remember. Okay, next. Now if we look into autosomal recessive disease, clear with X-linked recessive beta? X-linked only two disorders you will get for sure compulsory in your exam. What are those two? Color blindness and hemophilia. We have discussed the type of disorders and the proteins which are mutated because of which the disease is causing, and we have also discussed all the possible set of crosses for that particular diseases. Now let us focus on autosomal recessive disorders which are following Mendelian pattern of inheritance. So the very first disease is sickle cell anemia. First let us understand sickle cell anemia. Before that you have to be clear with the structure of hemoglobin. What is hemoglobin ma? Hemoglobin is a tetramer, right? It is a tetramer globulin protein. It is a tetramer globulin protein, okay, which has 2 alpha chains and 2 beta chains. It has 2 alpha chains and beta chains. Any of the structural disintegration in the hemoglobin will affect the functions of the hemoglobin, right? Any of the effect in the structure of the hemoglobin will affect what? functions of the hemoglobin. What is the function? Basic function of the hemoglobin? It will carry the oxygen from lungs to all the organs and tissues in the body, right? It will carry oxygen from lungs to all the tissues and organs of the body, and it will collect the carbon dioxide from all tissues and organs towards the lungs for purification. So yes, you will see oxyhemoglobin, right? Whenever the hemoglobin is reduced, oxygen tension levels in the organs will increase. Okay, oxygen tension levels in the body will increase and they will be anemic. They will be what ma? Anemic. Now if you look into this sickle cell anemia disorder, this is caused because of the mutation at the 6th position. This is caused because of the mutation at 6th position in the beta globulin protein. At what position? At 6th position in beta globulin protein, and this is seen in the chromosome level. You have to remember the chromosome number and you have to remember which position the globulin protein is affected, whether it is alpha or beta. For sickle cell anemia, it is beta globulin protein affected at 6th position and the chromosome 11. And this is an autosomal recessive disorder. Of course, you remember it for sure. The sickle cell disease is controlled by single pair of alleles. Okay, it is controlled by single pair of alleles, that is, so this alleles can be in 3 combinations beta. This is normal combination, this is heterozygous condition, and this is homozygous condition. As this is recessive, now you tell me in which condition the disease is seen. The disease is obviously seen in the homozygous condition. Okay, homozygous condition only the disease is seen. It is latent in the heterozygous condition. Even in the sex determined, in the X-linked recessive traits also, carrier mothers are latent, okay, wherein the disease is unexpressed, not distinguished in the heterozygous condition, and this is normal set of alleles where disease is not seen. Disease is only seen in the homozygous set of genes, homozygous set of genes which are affected, right? Homozygous set of alleles in a gene which are affected. They show diseased phenotype, okay, they show diseased phenotype, HBS, okay. Hemoglobin which is affected with sickle cell anemia in the both alleles only will show the disease. As a result of this alteration in the shape of RBC, okay, the normal shape of RBC is what ma? We have read in Cell the Unit of life, no? What is the normal shape of RBC? It is round and biconcave, right? Round and biconcave. This round and biconcave shape of the RBC will be modified to sickle elongated shape like this. It is modified to sickle elongated shape which will block the blood vessels. This will block the blood vessels and it will not carry the oxygen to the organs of the body. So thereby oxygen tension levels will be increased. Clear ma? So this you have to remember. Now let us understand how the mutation is happening at the 6th position of beta globulin chain. If you look clearly, the deficiency of RBC is due to replacement of what amino acid ma? Glutamic acid with valine. At which position? At 6th position. So if you look into it clearly, from DNA what is produced? mRNA, and this process is called as what ma? Transcription, right? Transcription. At 6th position, when CTC is there, complementary to this, what is coded in the mRNA? GAG is coded in the mRNA, okay. And this, when this is translated to amino acid, when this is translated to amino acid, glutamic acid is formed at the 6th position. But then just by mutation of single nitrogen base during DNA replication, which is CAC, during transcription mRNA is copying as GUG. mRNA is copying as what ma? GUG, and during the time of translation this is coded to what amino acid? Valine, which results in the change in shape of RBC. This results in what? Change in the shape of RBC. Questions appeared are, they will be asking you which globin, that is beta globin chain, and which position of that, 6th position, which amino acid is changed to what, glutamic acid is changed to valine, which leads to sickle cell hemoglobin, and they will be asking you GAG is coding for what, glutamic acid, and GUG is coding for valine. That's it beta. This is what you have to remember from the disease sickle cell anemia. Now you see, base substitution is happening at the 6th codon of beta globin gene from GAG to GUG. Next, the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing change in the shape of RBC from biconcave disc to sickle shape like structure. Next, if you look into the disease, second autosomal recessive disease, phenylketonuria. This is an inborn disorder. Okay, this is an inborn disorder only. Like you know, phenylketonuria is caused when phenylalanine is not transforming or changing to tyrosine. Phenylalanine to get transformed to tyrosine, there is an enzyme which is catalyzing the activity, that is phenylalanine hydroxylase. That is what, ma? Phenylalanine hydroxylase. If mutation occurs for this one enzyme, there are so many set of symptoms caused just because of the mutation of this single enzyme or protein. Now let us understand it more better. Phenylketonuria is an inborn error metabolism. This is an inborn error metabolism that results in decreased metabolism of amino acid phenylalanine. Okay, and this is because of the absence of the enzyme phenylalanine hydroxylase. This is inherited as what, ma? Autosomal recessive. The affected individual lacks an enzyme that converts amino acid phenylalanine to tyrosine. What is the name of that enzyme? Phenylalanine hydroxylase. Phenylalanine accumulates and is converted to, as this is not converted to tyrosine, this is accumulated as phenylpyruvic acid. Okay, it is accumulated as what, ma? Phenylpyruvic acid. This is accumulated as phenylpyruvic acid and other derivatives. Accumulation of this in the brain results in the mental retardation. If it is accumulated in the brain, it is resulting in what? Mental retardation. This also causes decreased absorption by kidneys, and pure absorption by kidneys is resulting in frequent micturition in those affected individuals. Now if you look into the multiple symptoms that are caused, the multiple symptoms which are caused because of the error in one enzyme. Okay, I hope it is clear, right? Phenylalanine has to be converted to tyrosine, and that is mediated by the enzyme phenylalanine hydroxylase. Absence or mutation in that one enzyme, phenylalanine is stored in the form of phenylpyruvic acid. This will result in the affecting in multiple terms like poor absorption in kidneys, mental retardation if phenylpyruvic acid is stored in brain. If it is stored in brain, this results in mental retardation. Then frequent micturition because of pure absorption by kidneys, then skin rashes and hyperactivity, and this we will also learn under hyperactivity. So these are all the symptoms of phenylketonuria. Okay, this is also studied under the phenomena pleiotropic. Okay, which are the exceptions of Mendelian inheritance? What is that? Pleiotropic. This is also studied as an example of pleiotropy because absence or mutation of the one enzyme phenylalanine hydroxylase is resulting in these many symptoms. Just one protein or one gene absence is resulting in these many symptoms, where one gene is coding for more than one character or trait, right? So that is the reason we are studying the disease phenylketonuria under the example of pleiotropy as well. Okay, this is the disease. One more disease we have to look into, and the third autosomal recessive disorder where we need to focus is thalassemia. Okay, what is it, ma? Thalassemia. Previously sickle cell anemia, it is because of the change or base substitution of one base, one nitrogen base. So it is called as qualitative. It is called as what? Qualitative. Thalassemia is a quantitative disorder. It is what, ma? Quantitative. Wherein we already learnt hemoglobin is a tetramer with 2 alpha globulin and 2 beta globulin subunits. Okay, thalassemia is caused when there is more synthesis of alpha globulin and reduced synthesis of beta globulin, which is said to be beta thalassemia. And if there is reduced synthesis of alpha globulin, there will be more synthesis of beta globulin, and abnormality of hemoglobin will again result in the disorder. So this is quantitative. Okay, it is based on the number of production of alpha and beta subunits. Now if you see thalassemia, it is an again autosomal recessive disorder of the blood which is transmitted from parents to offsprings when both the parents are unaffected carriers. As this is autosomal, if two parents are unaffected and acting as carriers, definitely the generation or the offsprings of those parents are affected. Next, the defect of mutation that causes thalassemia could either be due to mutation or deletion. It can be either due to mutation or deletion. Mutation or deletion of alpha globulins will decrease the number of alpha and increase the number of beta globulins in the hemoglobin. Decreased or mutation or deletion in the beta globulins, it will increase the synthesis of alpha globulins in hemoglobin protein. This causes the formation of abnormal hemoglobin molecules. Okay, what does this cause? This causes the abnormal hemoglobin molecules resulting in anemia again. This any change or disruption of the structure in the hemoglobin will affect what, ma? Function of the hemoglobin, resulting in anemia, which is characteristic of the disease. The characteristic of the disease thalassemia is again the same, that is anemic. Next, this is quantitative. Sickle cell anemia is qualitative. These are of two types, that is alpha thalassemia and beta thalassemia. Now we will study like what genes are affected in alpha thalassemia and beta thalassemia, what chromosomes are involved in alpha and beta thalassemia. If you look into alpha thalassemia, production of alpha globulin chain is affected. Production of alpha globulin chain is affected. Alpha thalassemia is controlled by two closely related genes, that is HbA1 and HbA2 from chromosome 16. Okay, chromosome 16 has one short arm and one long arm. If you remember clearly, short arm is said to be P arm, long arm is said to be Q arm, right beta? So on short arm, two alleles, that is two closely related genes HbA1 and HbA2 are affected, and this results in the reduced synthesis of alpha globulin. Next, due to mutation or deletion of one or more of these four genes, okay, these two are genes, okay, these two present on the homologous chromosomes means four genes. Any mutation in these four genes, the more genes are affected, less alpha globulins are synthesized. Effect in the more genes will decrease the synthesis of alpha globulin chains. More the decrease in the alpha globulin chains, you can see increase in the beta globulin chains. Thereby the structure of hemoglobin will be affected, and this will lead to oxygen dissociation curve disruption. Oxygen dissociation curve will be disrupted because of alpha thalassemia. Very important point you have to remember. What will be affected because of alpha thalassemia? Oxygen dissociation curve will be disrupted. Okay. Next, beta thalassemia, production of beta globulin will be affected. If it is alpha thalassemia, production of alpha globulin will be affected. If it is beta thalassemia, production of beta globulin chain will be affected. Beta globulin production is controlled by single gene called as HBB on the chromosome 11. Okay. Thalassemia, alpha thalassemia is on chromosome 16, that is in the short arm, P arm, right? And beta thalassemia is on the chromosome 11. Mutation in one or both the copies of this gene causes thalassemia. In one or both the copies of the gene, any of the copies are affected, then it will lead to beta thalassemia. Beta thalassemia will first affect, or they will bind to cell membrane of RBC. Cell membrane of RBC will be disrupted or affected. Alpha thalassemia will affect oxygen dissociation curve. Beta thalassemia, it will affect or disrupt the cell membrane of RBC. Okay. Next, if you look into the last autosomal recessive disease, which is cystic fibrosis. Most of you might be remembering this disease as 65 roses, but then remember it as autosomal recessive disorder. This is what autosomal recessive disorder. It is caused by mutation of the gene on chromosome 7. You have to remember the chromosome number. Okay, it is because of the mutation in the gene chromosome 7. Okay, cystic fibrosis is a genetic disorder that affects mostly lungs. Okay, also pancreas, livers, kidneys and intestine. Why is it affecting the lungs? As the effect is on lungs, definitely they will be difficult to breathe and coughing up the mucus as a result of frequent lung infections. The mucus traps the germs and leads to infections also. It can also cause severe lung damage like cysts which are filled in the lungs because of the mucus. Okay, because of the excess mucus, cysts are formed in the lungs. Because of that, continuous coughing, and they will cough the mucus and there will be great difficulty in breathing. So yes, cyst means fluid filled sacs, fibers means scar tissues. Okay, this is what cystic fibrosis, which is an autosomal recessive disease. They will affect lungs. Okay, and they will cause cysts or fluid filled sacs in the lungs. Clear? Next, now we will look into chromosomal disorders. What we look into? Chromosomal disorders. If you look into chromosomal disorders, these disorders occur because of the absence or excess or aberrant organization of one or more chromosomes. This is because of the absence or excess or aberrant organization in the chromosomes. These are of two types again, aneuploidy and polyploidy. These are of two types, what aneuploidy and polyploidy. Aneuploidy is caused because of non-disjunction of the pair of chromosomes. It is caused because of what non-disjunction of pair of chromosomes. What does it mean? It means that two chromosomes during anaphase, anaphase 1, okay, these are homologous chromosomes, they will be segregated in the anaphase 1, right? Because of not or inappropriate segregation, the chromosomes are segregated as such. The chromosomes are segregated as such. Because of this segregation, okay, because of this segregation, aneuploidy occurs. This is called as non-disjunction of pair of homologous chromosomes during anaphase. Either of the chromosomes will be moving to the opposite poles in an abnormal way, in an aberrant way, because of which either excess chromosomes will be formed or less chromosomes will be formed. If the excess chromosome is formed, like 2n plus 1, as we have seen here, one more chromosome is formed along with this 2 chromosome, which is the condition is what 2n plus 1. Excess chromosome is formed, which is 2n plus 1. This condition is said to be trisomy. This condition is said to be trisomy. So, when the disease is occurring, it is because of inappropriate chromosomal segregation during anaphase 1 of meiosis. Okay, it is because of inappropriate chromosomal segregation. If extra chromosome is transferring to the opposite pole, then it is trisomy 2n plus 1. This is because of addition of one extra chromosome. Monosomy or nullisomy. Monosomy or nullisomy. This is caused by deletion of one or more or both the chromosomes of the homologous pairs. Okay, either it can be 2n minus 1, which is said to be monosomy. Only one chromosome you can see on the pole. Okay, if two chromosomes are moving towards the opposite pole or two chromosomes are not at all there, then it is 2n minus 1, which is nullisomy. Okay, polyploidy. It occurs due to failure of cytokinesis after karyokinesis. After karyokinesis, cytokinesis will not happen, which is said to be cytokinesis is not followed by karyokinesis, then polyploid condition occurs. So, aneuploidy and polyploidy comes under chromosomal disorders. Under aneuploidy, we have to know about trisomy, monosomy and nullisomy. Trisomy condition is 2n plus 1, monosomy is 2n minus 1, nullisomy is 2n minus 2. Okay, next. So, yes, chromosomal disorders are of two types beta. If you look into human nucleus or human chromosome set, how many set of chromosomes we have? We have 23 pairs of chromosomes. We have how many? 23 pairs or 46 chromosomes. 46 chromosomes, of which 22 pairs are autosomes. 22 pairs are autosomes. One pair is allosome. One pair is allosome or sex chromosome. Okay, in females it will be XX, in males it will be XY. Okay, now if you see autosomal involving 22 pairs of autosomes, allosomals or sex chromosomes occurring X and Y or XX chromosomes. Okay, so aberration or chromosomal mutation in any one of this, be it autosomes or allosomes, will lead to chromosomal disorders. Based on that, from our NCERT, we have to focus on three diseases of under chromosomal disorders. Those are Down syndrome, Klinefelter syndrome and Turner syndrome. Down syndrome, if you look into, this is a genetic disorder. This is a genetic disorder caused when abnormal cell division results in an extra copy at which chromosome? At 21 chromosome, which is an autosome. Okay, this is discovered by John Langdon Down. Okay, who saw these characteristics for the first time. It is named after this scientist as Down syndrome. In males if you consider, the gene type will be 45 plus XY as this is in the autosome. In females it is 45 plus XX. You have to remember this for sure. Okay, this is a genetic disorder. This occurs because of the trisomy in the chromosome number 21. Down's syndrome. Okay, if you see the karyotype of Down syndrome clearly, karyotype of the Down syndrome, 21st chromosome. See here, 21st chromosome has trisomy, rest all chromosomes are diploid. Okay, only this one chromosome is showing the condition of trisomy. This is because of non-distinction of homologous chromosomes during which phase of cell division beta? During anaphase. Okay, next, what are the symptoms of Down syndrome? The symptoms of Down syndrome will include, they show typical facial features. Wherein the eyelids will be downward. Or epicanthal skin fold will be seen at the eyelids. Okay. Second, the face is typically flat with round flat nose and the tongue is protruding out all the time, okay, because of the frequent salivation, okay. Next, mental retardation is seen in the kids who are having Down syndrome. Lately, poor skeleton development is observed, smaller skull, and their palate is arched. Their palate is arched. They have flat hands with more creases. They have flat hands with more creases on the palm. So yes, what are the symptoms of Down syndrome? You have to remember, many a times the questions are asked in match the following, wherein typical facial feature like downward slanting of eyelids, second is round flat nose, and tongue is always protruding out. Major symptom is mental retardation, then poor skeleton development, lastly smaller skull and their palate is arched. They have flat hands with the more creases, okay. In NCERT there is best diagram of that, just look into it beta. Next, Klinefelter syndrome. Klinefelter syndrome, if you look into, this is observed in males. Klinefelter syndrome you will see in what? Males, wherein the chromosome number is more, or the trisomy is appeared at XY chromosome. Trisomy is appeared where ma? At X chromosome, XY chromosome. The trisomy is appeared over here. Klinefelter syndrome is a genetic condition in which boy is born with extra X chromosome. The boy is born with extra X chromosome. The extra copy of X makes karyotype of the condition 44 plus XXY. As the trisomy is there in the XY chromosome, the extra copy is in XY, okay. That is why the genotype which you observe is 44 plus XXY. If it is Down syndrome in males, it is 45 plus XY. It is what ma? 45 plus XY. Now if you look into the symptoms of Klinefelter syndrome. The symptoms of Klinefelter syndrome, which include development of breast, which is said to be gynecomastia. The males who are affected with this disease, the breast will be developed, which is called as gynecomastia condition, and these individuals are tall and thin. They are sterile and they cannot generate or produce sperm. Spermatogenesis is absent in the males who are affected with Klinefelter syndrome, and they cannot develop the secondary sexual characters of males, and such individuals are sterile. They usually have normal intelligence. Their intelligence is not, they have normal intelligence. They don't have mental retardation or abnormal intelligence. Their secondary sexual characters are affected by possessing gynecomastia. Second thing, they are sterile and they are poorly developed sexual characters. Lastly, spermatogenesis is absent and such individuals are sterile, okay. Next, Turner syndrome. Turner syndrome is seen in females, okay. Do remember children, Klinefelter syndrome is observed in males, Turner syndrome is seen in females, wherein you will observe 45 plus XO condition all the time. You will observe what? The monosomy in the XX, okay. One X chromosome is absent, because of which there are many characteristic features. Actually it is not 45, it is 44 plus XO, okay. This is 44 plus XO condition, wherein one more X chromosome is absent. There is only one X chromosome in the females who are affected with Turner syndrome. This is genetic condition in which female is partially or completely missing an X chromosome. The lack of one X chromosome in females, karyotype you can see like this, 44 plus X, only one X chromosome will be there. What are the symptoms for Turner syndrome? Symptoms for Turner syndrome. Yes, females with Turner syndrome are sterile. Females with Turner syndrome are sterile. Their ovaries are rudimentary, Okay, they have inactive or not well synthesized ovaries which are rudimentary. They lack secondary sexual characters again, and they have short stature, webbing of neck and low posterior hairline. These are the symptoms you will observe in Turner syndrome. So yes, students, what we have discussed today, the major concept from the chapter Principles of Inheritance and Variation, that is Mendelian disorders and chromosomal disorders, right? From Mendelian disorders, you have to focus mainly on X-linked recessive disorders. Those are color blindness and hemophilia, and all the 4 set of combinations where we have discussed. The pattern or the percentage of disease you will observe in the F1 generations. That is important. And from autosomal recessive disorders, 4 diseases you have to focus on. Those are cystic fibrosis, thalassemia, sickle cell anemia, and one more disease, that is phenylketonuria. Lastly, from chromosomal disorders, what you need to focus on? You have to focus on Down syndrome, which is because of the trisomy of 21, then Klinefelter syndrome because of the trisomy of XY chromosome in males. Lastly, Turner syndrome, which is because of monosomy in the X chromosome, only one X chromosome is female, and this you will see in females. Turner syndrome is seen in females, Klinefelter in males. This is the end of discussion. Coming up class, we will discuss about pedigree analysis. Thank you.